I'm going to talk about stress in relation to welfare and start by saying what do we mean by the term stress. And I would emphasize that there's only one biology. The same biological principles apply to humans and to non-human animals, and just with other terms like health and welfare, stress means the same for every living organism. So it means the same for a human or for a trout or for a moth. And for most people, stress implies the effects of a challenge to the individual that disrupt homeostasis and result in adverse effects. And if you have a stimulus which just activates energetically cheap control mechanisms and doesn't cause any negative effect, then in general people wouldn't use the word stress for that. And if there are stimuli whose effects are beneficial, they are not normally called stressors by most of the public or most scientists. Now, stressors can be physical effects or mental effects, and some of them will act on cells, of course. Any organism can be stressed, but we do have a lot of discussion about a particular mechanism, and that's the HPA axis, the hypothalamic pituitary adrenal cortical axis, because that's involved in some important responses of animals to difficult conditions. But that's also involved in lots of other areas of life, generally speaking, we shouldn't limit the word stress to one system. So stress is an environmental effect on an individual which overtaxes control systems and results in adverse consequences, eventually reduce fitness. If there are impacts of the environment which have no negative consequence, then they're really better called stimuli rather than stressors. Now, stressors sometimes have an effect which is at a different time from the time that they themselves act. So there may be a delay before consequences. We know that early negative experience can have effects in later life. And we also know that effects during the life of parent individuals can negatively affect the offspring. The most obvious thing is undernutrition in utero can have later consequences, but there are also effects which occur by an epigenetic route, that is, the consequence of the stimulus is to change something in the genetics of the offspring and maybe further generations. For example, when piglets are born from sows fed high fibre diets in early pregnancy, these piglets were less aggressive prior to weaning than if the sows had had a less good, not high fibre diet. Another example is when sows were lame during pregnancy, their piglets had more skin lesions, that is a result of fighting by the piglets after birth. So these then are effects which are occurring a long time after the initial effect. And the general message is that it's now clear that there can be negative environmental impacts on male mammals during sperm production and female mammals during egg production, which can have negative epigenetically controlled consequences for their offspring. So an example of that is that when boars were kept either in an enriched pen or in a crate, which is an impoverished environment, during their production of sperm, the offspring of these boars were subject to a differential amount of fighting according to the environment that the fathers had lived in just at the time that the sperm were being produced. So that if the boars had been in a good environment, an enriched pen, then the offspring showed less fighting, less skin lesion. Now, the negative environmental impacts can clearly occur at the cellular level, and very often the first effect of an environmental factor is at the cellular level, and this then is referred to as stress on these cells and as a consequence on the organism. So, for example, water deficit can disorder protein mechanisms in cells, preventing normal function and stressing the cell and stressing the organism. But if you have a consequence of a cellular response which is a positive one, then it would be better not to call it stress, even if you thought at the outset that it might be a negative experience. So high temperature leading to heat shock protein production can cause activation of an endoplasmic reticulum chaperone, which leads to refolding of proteins that had become misfolded during life. So that's a positive effect. The refolded proteins are going to function better than the misfolded proteins which had been there before. So this is an effect of a short period of high temperature, which is an adaptive response with good consequences for the individuals affected. So again, I would say that the effect is not stress. Welfare is a term this is the other major term which we're discussing here, is a term which can be used for any individual animal, not for plants, not for microorganisms or inanimate objects. There's got to be a nervous system acting for the word welfare to be used. And the welfare of an individual is its state as regards its attempts to cope with its environment. And all individuals, especially those with a high-level brain, sentient animals, including humans of course, have a wide range of coping mechanisms, including those which deal with pathology, so the things which are influencing health, and including high-level functioning in the brain, positive and negative aspects of the mechanism, which lead to feelings, positive and negative feelings. So health and feelings are important parts of welfare. Now, there's a wide range of measures of stress and welfare. The measures of stress are of negative effects, but welfare will include positive effects as well as negative effects. And there are some direct measures which indicate how good or how poor the welfare is at any particular time. And some of these are showing what's happening in the short term. Others are giving information about much longer periods. And then there's another group of measures which are used by animal welfare scientists which give information about what animals need, how important are particular resources to them. Measures of stress indicate likely negative impact. And of course, whenever there is stress, the welfare is going to be poorer than when there isn't stress. And overall, the sentient animals, which we're mainly concerned with in terms of farm animals, pet animals, laboratory animals, they have sophisticated systems for controlling their interactions with the world around them. And where those can act effectively and the individual has control, the welfare is better than when the individual doesn't have control. So if you can give animals better control of their environment, and that applies to humans as well as other animals, then the welfare is generally better than if there's lack of control of the environment. And we can use the fact that individuals can select environments. Researchers can get information about what environments are selected. So when you give the animal environmental control, that very often is what we might refer to as an enriched environment, we can find out how strong the preferences are for particular aspects of the environment. We can find out what the animals need. So here's an example of such a study. This is when rats have a choice of flooring, a choice of a solid floor or a wire mesh floor. If they're active, then they will walk around on both. But if they want to rest, then they strongly prefer the solid floor to the wire mesh floor, the wire grid. And that you can find out how strongly they prefer it by getting them to lift a door in order to get to the resource, and you find that they will actually lift a heavy weight in order to get to a grid floor, in order to get to a solid floor and away from a grid floor in order to rest. So we can use that kind of study to find out what's important to the individuals. The direct measures, we very often need to interpret a physiological measure in the context in relation to perhaps the behavior which is going on. So heart rate gives useful information about an animal's predictions about what's going to happen to it, but you want to know where it is and where in order to interpret that. The same applies to cortisol and corticosterone. They're very useful measures, particularly of short-term difficulties, but as well as occurring in situations which are adverse for individuals, they also cortisol or corticosterone may also change during positive situations. So you get an increase in cortisol during courtship and mating, which of course are not negative things at all. So you need to look at the context when you are deciding whether you're going to use a particular measure. Glucocorticoid concentrations, you need to relate it to what's the baseline and what's the maximum you could get, and then to the context. Whenever there is unpredictability, whenever there is a lack of control because the environment is changing in an unpredictable way, that tends to be a difficult time for individuals. It's a difficult time for humans, it's a difficult time for other species, and sometimes it will very often lead to increases in cortisol, and sometimes in other effects, the well-known effects on gastric ulceration in rats, which were subject to unpredictable shocks. Was one of the early studies in this area. If you're looking at short-term problems, there's a whole range of different aspects to short-term problems. So supposing you're going to transport an animal for a certain distance, that will result in several impacts of the environment. There might be food deprivation, in which case you want to measure free fatty acids, beta-hydroxybutyrate, glucose and so on. If there's dehydration, you may need to measure the osmolality, the body fluid concentration, the total protein, individual proteins. If there's something like bruising, then you might want to measure enzymes like creatine kinase and lactate dehydrogenase. If individuals are subject to something which is frightening, then you have a range of physiological measures which are useful to measure. Even things like motion sickness have changes in physiology which can be measured. Inflammation, immunological responses will change the proteins like several different kinds of acute phase protein. Temperature can be measured and so on. So we have a wide range of different measures for laboratory animals and for any other animals which we use. And whenever individuals are subject to handling as part of a procedure, that in itself might cause the individual to show an increase in glucocorticoids. So corticosterone in mice increases according to the magnitude of the change when they are handled and taken out of their home cage and put somewhere else. So these are useful measures, and the commonest laboratory animal now is the zebrafish, and if you catch them in a net and take them out of the water, then you get a big increase in cortisol. So you need to know about those sorts of things in handling and managing animals and designing experiments. It's a whole range of things which have been done in research in these areas to find out what is the extent of the response of the animal to the treatment. So things like restraint, electric shock, separation from mother, being attacked, being in a fight, and so on. And even information about something which could be negative, like being exposed to the odour of a rat which has experienced shocks, will lead to adrenal cortex responses in the one which is smelling that. And these changes can damage the animal in some way. They can damage the brain, they can impair cognition, and so we need to know about those consequences. And we have other consequences which of negative things like depression, and depression occurs in all sentient animals in certain circumstances. We can find out what are the effects on particular cells in the brain, what are the effects on the functioning of the immune system. We know that a chronic adverse environmental impact can change learning and memory efficiency. It will damage neurons, alter the structure of the system in the brain, and it's quite clear that these are negative things for the animal. This is certainly a stressful situation for the animal. Whenever the immune system activity is high, that means they're working hard to try to deal with pathogens or something else which is happening negative. There are various corollaries to that which we can measure, and so there's a whole lot of things which can be measured which are telling us what's going on in the brain and in other parts of the body in trying to cope with difficulties. And there are different effects on the brain for different kinds of coping systems. And so some are short term, some are longer term, and we can measure these things. We know what is happening in the learning system, in the emotional parts of the brain. We can actually see what cortisol, for example, is taken up by cells in the brain. There are receptors in lots of different parts of the brain, and we know that the normal diurnal rhythm of cortisol is something which is actually quite important, because individuals who are caused very difficult conditions may lose that rhythm of cortisol. Also, going to the more positive side, we know something about what goes on when individuals are feeling pleasure. So you can find oxytocin levels increasing during a range of pleasurable events, and they have interactions with the other systems which are functioning. So we want to know what's happening in the brain with the positive and the negative in life. Oxytocin is associated with a range of other important things which happen during life. So how is this one biology approach relevant to stress and welfare assessment? Well, the main way that it is that humans have complex mechanisms for dealing with problems, and so do all other sentient animals. So brain mechanisms are very important in coping with the environment of the individual, and hence with the welfare of the individual. So if you have a particular kind of coping method, it will have some costs as well as some benefits. And these can be evaluated, and they need to be evaluated in determining what's the best strategy in life. And looking at these strategies that individuals have in trying to deal with problems which they encounter, or situations which are positive but where there might be problems, are things which can be measured directly. So, for example, supposing the individual is going to avoid and flee from a situation, that individual needs to consider the energy cost, the risks of mortality and injury, consequences for activities like feeding and reproduction, consequences for the amount of anxiety and depression, or the amount of pleasurable activity which might occur. All of these are things which can be measured and which we need to do in evaluating welfare and in considering the extent of stress. If here's a list of a whole range of coping responses which can be used by a very wide range of animals, where we can consider how much is going on which is causing unusual behavior, which may be harmful or maybe just something which we can measure. And so some of these are very sophisticated things, some of them are more straightforward things, but we can measure those things in terms of evaluating the welfare of individuals. And we can measure the welfare of humans, we can measure the welfare of all other species. So individuals use coping responses, and they may switch from one response to another, and that's an important subject area for animal welfare science. And in order to understand that, we have to understand motivation, have to understand what is involved in switching from one activity or one brain area of function to another. And so we need to use neuroscience techniques as well as behavioral techniques and physiological techniques. And overall, in evaluating stress and evaluating welfare, we need to put it in the context of the rest of the world. So we need to present the welfare of individuals as a component of the sustainability of systems, which is an important one for most people in the world. Obviously, we need to consider ethical arguments and the ethical methods of ethical discussion. And animal welfare scientists need to be able to present those results to the people who have to take decisions. So those might be lawyers, they might be politicians. We need to be aware of the place of stress and welfare research in the world in general. Okay, so these are some of the books which I've referred to where I've written about these things, and I've put a lot of references in this text, and here are the references which I've cited. So thank you very much.